Welcome friends for online session 2, class 12th, subject biology, sexual reproduction in plants, chapter 2. I am G.R. Chopade, PGT biology, JNV Sangli. So today we are going to discuss introduction, sexual reproduction. In last session we have seen a flower structure and male reproductive organ. Now today we will discuss flower, that is female reproductive organ, and the different pre-fertilization process. So already we have seen this, a flower. Okay, all these parts we have seen, a sepal and a petal. Okay, female reproductive part. So the female reproductive part consists ovary, swollen part, style and stigma. And inside the ovary, so there are ovules. Now first watch this video.
All of us. They are the most beautiful part of the plant, isn't it? You already know that flowers are not present only to add beauty to the plant. They are in fact the reproductive part of the plant. In the flower, stamen is the male reproductive part, while carpel is the female reproductive part.
So these are the parts.
Some flowers contain both male and female reproductive parts. Such flowers are called bisexual flowers.
Bisexual
For
flowers.
example, pea, China rose, etc. On the other hand, some flowers possess either the male or the female reproductive parts. Such flowers are called unisexual flowers. For example, corn, cucumber, etc. Now let us know about the different parts that make up a stamen and a carpel. A stamen is made of two main parts, anther and filament. The anther produces pollen grains. It is the pollen grain that contains the male gametes.
Two male gametes.
A carpel, on the other hand, is made up of three parts. The terminal part is called the stigma. The middle elongated part is the style, and the swollen bottom part is the ovary.
Three parts.
Inside the ovary, ovules are present. It is the ovule that has the female gamete called the egg cell. For sexual reproduction to take place, a male gamete is to be fused with the female gamete, thereby forming a zygote.
So this is the result of
Now for
sexual
that,
reproduction.
the male gametes have to reach the female gametes. For this, first a pollen grain carrying the male gametes is transferred from the stamen of a flower to the stigma of the same or the other flower. Such transfer of pollen grains is known as pollination. If the pollen grain of a flower lands on the stigma of the same flower, it is called self-pollination.
So this is self-pollination, same stigma.
But if the pollen grain of a flower lands on the stigma of another flower of the same or another plant of the same type, it is called cross-pollination.
So this is cross.
Pollination occurs with the help of different agents like wind, water, or animals. Once a pollen grain reaches the stigma of a suitable flower, a tube grows from the pollen grain. This tube travels through the style and reaches the ovary. The male gamete from the pollen travels in this tube and reaches to the female gamete present in the ovule.
So there are two male gametes reach to the embryo sac. We'll see that.
Ovule of the ovary. After that, the male and the female gamete fuse together to form the zygote. This process of fusion of male and female gametes is called fertilization. The zygote formed after the fertilization then divides several times to form an embryo inside the ovule. The ovule develops a tough coat and changes to a seed. The ovary, on the other hand, ripens and forms a fruit around the seed. At this time, the other parts of the flower, like the sepals and petals, fall off.
Everything is fell down.
So, a seed present inside a fruit contains an embryo, which is actually a baby plant. Each time a seed is planted in soil and provided appropriate conditions, it grows into a seedling, which then...
Okay, so this is how the complete sexual reproduction takes place. Now, so we'll continue with the first session. Now, gynosium consists of a single pistil, that is monocarpellary, or it may have more than one pistil, then it is called multicarpellary. When there are more than one carpel, so the pistil may be fused together, that is called syncarpus, or it may be free, that is apocarpus. So in 11th standard you have studied what is syncarpus and apocarpus. So this is a syncarpus and this is a apocarpus. Okay, so each here it is free and there are fused. Okay, now pistil has a three parts: stigma, style and ovary. So stigma serves as a landing platform for the pollen grains. तो pollen grains उधर land होते हैं. Style is an elongated slender part beneath the stigma, यानी below the stigma. Ovary a basal bulged या swollen part of the pistil, वो नीचे होता है, आपने diagram में देखा है. Our inside the ovary is the ovarian cavity या locule. Okay, उसके अंदर ovules होते हैं. So what are ovules? So ovules are developed from the placenta and is located inside that ovarian cavity or locules. So they are developed from the megasporangia. So megasporangia arise from the placenta. So megasporangia are referred as ovules when they are mature. So the number of ovules in an ovary may be one. The best example you can see here, wheat is there, paddy is there, mango is there. Or there may be many ovules in one, papaya, watermelon, yeah, orchids. Orchids has the smallest and largest number of seeds. Okay, a NEET mein questions aayenge. See that. This is how the placenta, on that placenta, here you can see a megasporangium, and this will become the ovule. Now, how it will become, that will study. So megasporangium or ovule. The ovule is a small structure attached to the placenta by a stalk, and that stalk here referred as a funicle. So the body of the ovule fuses with the funicle in the region called a hilum, that is the hilum. So hilum represents the junction between ovule and funicle. You see that diagram. So each ovule has one or two protective envelope that are called इंटुगुमेंट्स. यही इंटुगुमेंट्स आगे जाके सीड कोट बनते हैं. इंटुगुमेंट्स encircle the whole part except at the tip where a small opening is there, और उस opening को हम बोलेंगे micropyle या micropyle end. Opposite the micropyle end, उसके बिल्कुल opposite, there is a chalaja representing the basal part of the ovule, base and up. Okay, you can see here different. So nucellus, inside there is a nucellus, a mass of cells enclosed within the integuments. So cells of the nucellus have a abundant reserved food material. बहुत सारा food materials होता है. And located in the nucellus is the embryo sac or a female gametophyte. Now this is a micropyle end and this is chelagel end. So these are one or two integuments and this is a funiculus and here we'll get a hilum. Okay. So inside there is a nucellus and this nucellus will develop into a embryo sac or this is called a female gametophyte. You can see there is a chelagel end and there is a micropylar end. Okay. So inside different cells are there. We will see later. Now, so where from this is developed? This is developed from a Megasporogenesis. Now, so this is a what you call a typical ovule. I have shown here different types of ovule, but this one is a typical ovule, that is an anatropous ovule. See here a micropyle end is there, one or two integuments are there. So inside there is a embryo sac. This is micropylar end Opposite to the micropylar is chelagel end. Then there is a raphe also. So this is the hilum where the funiculus attached to the ovule, and funicule is the stalk. Okay, so this is orthotropous A1. So this is hemitropous. You can see that here the micropyle and funicle are parallel to each other. Here micropyle and funicle in the one line. Okay, here the micropyle is right angle to the funicle, so depends upon that position. So there are different names. Okay, but you remember only a anatropous ovule. Okay, so this is important one. Now this is what a embryo sac. Okay, now so process of formation of megaspores from the megaspore mother cell, yeah, MMC. Okay, so here you can see megaspore mother cell. Here there is a surrounding cell nucellus, micropylar end. So differentiate a single megaspore mother cell in the micropylar region of the nucellus. Okay, so it is a large cell containing a dense cytoplasma and a prominent nucleus. So the MMC undergo meiotic division. See, meiotic. And meiosis result in the production of 4 megaspore. First, megaspore diad will produce, then the megaspore tetrad will produce. So from one megaspore mother cell, 4 megaspores are produced in a linear fashion. So this process is called Megasporogenesis. Now, how that megaspore become female gametophyte? So out of 4, one of the megaspore is functional, especially which is present chelagel end, while the remaining 3 will degenerate. So therefore, out of 4 megaspores, 1, that is monosporic, only the functional megaspore develops into the female gametophyte, or also called embryo sac. So monosporic development का मतलब क्या है? 1 megaspore develop into 1 embryo sac. Out of 4, 1 will become functional. How it will develop? The nucellus of the functional megaspore divides mitotically to form, okay, nucleus. Nucleus of the functional megaspore divide mitotically 2 nuclei. Okay, first 2 nuclei which move to the opposite pole, forming the 2 nucleate embryo sac. पहला division हो गया. Then again 2 more, एक के बाद एक, 2 more sequential mitotic division result in formation of the 4 nucleate. Okay, then 8 nucleate stage. So 2 nucleus, 2 divide होंगे, 4 nucleus, 4 again divide होंगे, 8 nucleate stage of embryo sac is produced. So after the 8 nucleate stage, the cell wall produce. Okay, cell wall. So you can see here how it will produce. See, 2 nucleate, then 4 nucleate, then 8 nucleate. Now these 8 nucleate arrange themselves. See here. So 3 will go at the chelagel end and they become 3 antipodal cells. All are haploid. Then 3 will move to the micropylar end. Out of that, 1 is become egg and 2 will become synarchids. And these synarchids have a filiform apparatus, finger-like projection. So they guide the pollen tube. Very, very important function of synarchids. They guide the pollen tube entry into the embryo sac. And the 2 will remain in the center. So they are called polar nuclei. Then when the cell wall is produced, here the 3 cell become, here the 3 cell, 6 cell, but here only 1 cell is produced containing the 2 nucleus, and that cell is called central cell. And this will, after fertilization, see here 2 polar nuclei, 3 antipodal cells, 1 egg cell and 2 synarchids. So this is how the structure. So this embryo sac is called 8 nucleate and 7 celled. ये बहुत important question है, बार बार पूछेंगे. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus consists 2 synarchids and 1 egg cell. The synarchids have special cellular thickening at the micropylar tip called filiform apparatus. उनको क्या बोलेंगे? Filiform apparatus. Then these helps in guiding the pollen tubes into the synarchid. तो pollen tube हमेशा पहला enter करेगा synarchid में. 3 cells at the chelagel end opposite and are called antipodal cells. A large central cell रहेगा which has 2 polar nuclei. डायग्राम निकालना है जो मैंने पहला दिखाया और स्ट्रक्चर लिखना है. मोनोस्पोरिक 8 nucleate 7 celled embryo sac. Now once pollen grain is developed, male gametophyte, female gametophyte is developed, now ready for the next process, pollination. Transfer of pollen grains from anther to the stigma of pistil. Adaptation to achieve pollination, either in a types of pollination, autogamy, yes, cell pollination, Geitonogamy and genogamy, yeah, cross pollination. So there are 3 types of pollination. Autogamy, transfer of pollen grains from anther to the stigma of same flower. Here flower requires synchrony in pollen release and stigma receptivity. पोलन ग्रेन भी उसी समय रिलीज होना चाहिए जब स्टिग्मा मैच्योर होगा तभी पोलिनेशन होगा. Here there are chasmogamous flowers with exposed anthers and stigma. Best example is oxalis. या clistogamous flowers which do not open at all. Best example is comelina. So here 100% cell pollination या autogamy होता है. See here how that stigma pollen grain is received from the same anther, and here you can see these are chasmogamous flower and these are never open. They are called clistogamous flower. ये डायग्राम दिखाएंगे, ए और बी आइडेंटिफाई लेबलिंग के लिए आएगा. Okay. Now second one is Geitonogamy. Transfer of pollen grains from the anther to the stigma of another flower of the same plant. Plant एक ही है लेकिन ऊपर और नीचे फ्लावर है. पहले में फ्लावर एक था. Involves pollination agent. यहाँ पे एजेंट्स की जरूरी है, either wind या whatever it is. But genetically it is similar to autogamy या cell pollination because same plant. Okay, only flowers are different. Neighbor marriage इसको बोलेंगे. Okay, like this. Now third is genogamy. Transfer of pollen grains from the anther to the stigma of different plant of a same species. Plant अभी अलग है, flower तो अलग ही है. Plants are also different. It brings genetically different types of pollen grains to the stigma. Here, like that, A flower, B flower. So vice versa. Okay. But species are same. Now, to do cross-pollination, agents are required. Pollinators are required. So abiotic pollinators like wind and water. Best examples, yeah, biotic pollinators, many animals are there. Abiotic, best example, grass, a corn cob, that is maize, yeah, Vallisneria and Hydrilla. And in biotic, so many are there. Just watch this video. So this is insect pollination. Okay, see, bee, honeybees. They carry secrete for nectar, attracted by its vibrant color. They carry the stigma, pollen grains to the stigma here. Search of the nectar they are keeping inside. Okay, so this is honeybee, that is insect pollination. Now here you can see during the night, bat pollination. The bat also do pollination. So they will collect, see the flower nature. Okay. Now we will discuss one by one. Now pollination by wind is also called anemophily. Transfer of pollen grain by wind from one flower to another flower. What are the characters adaptation? Questions आएंगे. Flowers generally unisexual. Best example coconut palm, date palm, maize, many grasses या cannabis. The pollen grains are produced in large quantity, large number. Cannabis 5 लाख से ज्यादा pollen grains तैयार होते हैं. मेज 25 मिलियन पोलन ग्रेन्स तैयार करता है. पोलन ग्रेन्स आर वेरी स्मॉल, स्मूथ एंड ड्राई एंड लाइट इन वेट. हल्के रहते हैं. See that here the anthers, okay, anthers are very versatile, exposed, and when the air will come here, they will, here see corn cob, the stigma, okay, style will expose and very sticky, hairy structure. Gunpowder mechanism also shown by Euretica, that is anther burst suddenly, so the pollen grain will spread in the air, Euretica. So the winged pollen grains are also produced like pines, and which are flow around 1300 kilometer away from the parent plant. Flowers here are generally colorless, no color, odorless, nectarless, क्योंकि wind है. Flowers produce above the foliage, यानी leaves के ऊपर exposed रहते हैं, above the foliage. Both stigma and anthers are versatile, यानी ऐसे हिलने लगते हैं, so that to catch, and here the stigma is hairy and feathery. So this is the characteristics of wind pollinated flowers. Example मैंने दिखाया. इधर भी देख सकते हैं, flowers are unisexual, male flower, female flowers, या anthers are versatile. Now second one is hydrophily, that is pollination by water, या water pollinated plants. It is common among monocot genera. All aquatic plants are not pollinated by water, या hydrophilous. Some hydrophytes, anemophilous, example Potamogeton, या Myriophyllum, or some are entomophilous, that is insect pollinator, Alisma and lotus. See, lotus is even present in the water, but flowers are exposed, and so it is attractive. So remember this one, only monocot genera. Now, flowers may be pollinated inside the water, that is hypohydrophily, below the surface of water. Joastera, Ceratophyllum is the best example. Or at the surface of water, epihydrophily. बेस्ट एग्जाम्पल वेलिस्नेरिया, या टेप ग्रास भी बोलेंगे, या रिबन वीड भी बोलेंगे. सो दिस इज हाउ इट इज शोन ए वेलिस्नेरिया. नाउ विल सी वन बाय वन. पोलिनेशन बाय वाटर. इन वेलिस्नेरिया ये पूछेंगे, फीमेल फ्लावर्स रीच द सरफेस ऑफ वाटर बाय ए लॉन्ग स्टॉक and pollen grains are released onto the surface of water, so the water will carry, pollination takes place. In seagrass या जोस्टेरा, female flowers remain submerged, वो ऊपर नहीं आते हैं, and the pollen grains are released inside the water. So here pollen grains are boat shape, and here the pollen grains are needle shape, क्योंकि इनको sink होना पड़ता है, इधर float होना पड़ता है. Pollen grains are long and ribbon like, yeah, needle like. Both pollen grains are protected from wetting by a mucilaginous covering. क्योंकि they remain in the water, so if the mucilage coat is not there, the pollen grains will become wet and they start decaying. तो उसको prevent करने के लिए दोनों pollen grains में क्या है, mucilage. बाकी का similar to the wind, the flowers are colorless, odorless, okay. Pollination by insects, entemophily. Again we have seen in the video, here also in the different flower nectar. So flowers are large, colorful, fragrant and rich in nectar. इधर modifications भी होते हैं, corona in fashion flower, या petalite bracts in bougainvillea. They provide rewards to the animals. Pollen grains are sticky यहाँ पे, spiny and very rough होते हैं. Most of the flower are pollinated by bees. 80% flowers are pollinated by bees. So they use ultraviolet radiations for the detection and observations. So these are the characteristics of insect pollinated. Now come to the Ornithophily, that is pollination by birds. Sunbird, hummingbird, crow, या bulbul, so many. Example, coral tree, callistemon, या bottle brush, Butea monosperma, bombax, that is silk, cotton, all these trees. Characters क्या है flower के? Flowers secrete abundant, more watery nectar made of sugar, sugar ज्यादा होता है. They are very brightly colored but scentless, smell नहीं होती है, color ज्यादा होता है. Floral parts are leathery and funnel shape, leathery and funnel shape, ऐसा funnel shape होता है. Okay, see, pollen grains are sticky. Okay, see here certain example. ये है bottle brush. Okay, ऐसा ये है ब्यूटिया, ऐसा आपको नजर आएंगे. Now, the fourth one is Chiropterophily या Chiropterophily. The pollination are carried out by bats. Characters इनके flowers are dull colored होते हैं क्योंकि ये रात में होता है pollination, but strong fruity color. दूर से ही bat पहचान लेता है. Abundant nectar and pollen grains, सबसे ज्यादा होते हैं pollen grains. Flowers are large and stout, यानी ऐसा झूमते नहीं है, खड़े हो जाते हैं. बेस्ट एग्जांपल एडेंसोनिया या बहुबा ट्री, एक इसमें 2000 स्टेमन्स होते हैं. या काइगेल या पिन्नाटा, वो सुसेज ट्री बोलते हैं उसको, या एंथोसेफेलस दैट इज कदम ट्री. ये सभी आर पोलिनेटेड बाय बैट. ऑलरेडी मैंने वीडियो आपको दिखाया है. दिस इज हाउ द फ्लावर्स आर हियर इवन हैंगिंग. ओके, हियर द बैट इज पोलिनेटेड. ओके, नाउ सम स्पीशीज शो स्पेशल कैरेक्टर. They provide floral revites, providing the safe places to lay eggs. For example, the tallest flower, Anemophily, 6 feet. Here, the moth and plant yucca. Okay, moth and plant yucca. So, moth deposits its egg in the locule of the plant yucca. Okay, so here you can see the longest, tallest flower, 6 feet. Okay, and this is yucca, and inside that you can see, so that is a moth. It will use that to lay the egg, and during that the pollination takes place. So this is one kind of symbiotic relationship. You will study that one in even next chapters. There are also outbreeding devices. See here, stigma का height देखो और anther का position देखो, ऊपर है. इधर stigma नीचे है, anthers ऊपर है. या मेल फ्लावर अलग होगा, फीमेल फ्लावर अलग होगा. आउटब्रीड यानी टू प्रिवेंट सेल्फ पोलिनेशन और इनवाइट क्रॉस पोलिनेशन. ऐसे डिवाइसेस को हम बोलेंगे आउटब्रीडिंग डिवाइसेस. वेरी वेरी इंपॉर्टेंट. एडेप्टेशन फॉर क्रॉस पोलिनेशन डिवाइसेस टू डिस्करेज सेल्फ पोलिनेशन एंड एनकरेज क्रॉस पोलिनेशन. उसमें डाइकोगैमी, pollen release and stigma receptivity are not synchronized. Self pollination synchronization होना चाहिए, लेकिन क्रॉस पोलिनेशन के लिए वो अलग अलग समय पे होंगे क्योंकि वो दूसरे फ्लावर में तैयार रहता है. Either the pollen is released before, उसको बोलेंगे प्रोटेंडरी कॉटन, यानी पहला वो रिलीज होगा, मैच्योर होगा. और स्टिग्मा बिकम्स मैच्योर या रिसेप्टिव, उसको बोलेंगे प्रोटोगाइनी, दैट इज सोलैनम, मच बिफोर द रिलीज ऑफ पोलन ग्रेन. ये टेक्स्ट बुक में टेक्निकल वर्ड्स नहीं है, बट दे विल आस्क इन नीट एग्जाम. सो दिस इज द फर्स्ट डिवाइस, डाइकोगैमी. दूसरा डिवाइस है हेरकोगैमी, मींस एंथर एंड स्टिग्मा आर प्लेस्ड एट डिफरेंट प्लेसेस. मैंने डायग्राम में दिखाया, अलग अलग. एंथर ऊपर होगा तो स्टिग्मा नीचे होगा, स्टिग्मा ऊपर होगा तो एंथर नीचे होंगे, तो सेल्फ पोलिनेशन नहीं होगा, दूसरा पोलन ग्रेन आना ही है. बेस्ट एग्जांपल हिबिस्कस में देखा है. तीसरा है सेल्फ इनकम्पैटिबिलिटी, वेरी इंपॉर्टेंट. ये जेनेटिकली कंट्रोल्ड है. इट प्रिवेंट सेल्फ पोलिनेशन फ्रॉम फर्टिलाइजिंग द ओवल्स बाय इनहेबिटिंग द पोलन जर्मिनेशन. अपने ही पोलन जर्मिनेशन को होने नहीं देता है. एप्पल में पिटूनिया इज द बेस्ट एग्जांपल. फोर्थ है हेट्रोस्टाइल, दैट इज द लेंथ ऑफ स्टाइल इज डिफर्स इन फ्लावर. लॉन्ग स्टाइल, शॉर्ट स्टेमन, और vice versa, long stamen, short style, like that. Example, prime rose. और जो लास्ट डिवाइस है डाइक्लाइनी, दैट इज यूनिसेक्सुअल फ्लावर्स. आपके किताब में यूनिसेक्सुअलिटी, मैच्योरिटी टाइम इज डिफरेंट, नो सिंक्रोनाइजेशन और सेल्फ इनकम्पैटिबिलिटी ऐसा आपको दिया है. ओके, नाउ वंस द पोलिनेशन टेक्स प्लेस, हियर the pollen grain, either it is a two-celled, that is generative cell and vegetative cell, deposited on the stigma, that is pollination. Then the pollen grain germinate, produce pollen tube, that carry the male gametes. So two male gametes are there which reach to the embryo sac, and inside the embryo sac they release the male gametes as sperm cell, and here the two fusion takes place. One is produced zygote, another is produced endosperm. So therefore it is called double fertilization. Now how it will take place? Now pollen grain germinate on the stigma to produce a pollen tube through one of the germ pore. ये क्या दो germ pore होते हैं? So pollen tube grows through the tissues of the stigma and style and reaches the ovary. So the generative cell divides and form a two male gametes. आफ्टर जर्मिनेशन या बिफोर जर्मिनेशन. अगर बिफोर जर्मिनेशन हो गया 3 सेल स्टेज बोलेंगे, जर्मिनेशन के बाद हो गया तो 2 सेल स्टेज पोलन ग्रेन बोलना है. It enters the ovule through the micropyle and synarchids, okay, because there are filiform apparatus वो guide करते हैं. Okay, see here. So micropyle से end होगा, then here they will release synarchids, one to the sperm cell just before fertilizing the egg cell, and here sperm cell just before the fertilizing central cell. So these are polar nuclei, and here is a egg cell. Okay, तो इसी को हम बोलेंगे double fertilization. Again, if you refer there, okay, so there may be porogamy, chalezogamy, okay, ऐसा आपको आता है, या chalezogamy, chalezolent, porogamy, that is through the pore, ऐसा आपको मिलेगा. Now, so if you want to do a hybridization, that is artificially crossing the different species to combine desirable character to produce superior varieties by selection of desired parents, तो क्या करना है? Emasculation, cross pollination, bagging technique करना है. This will comes also for practical. Only the desired pollen grains are used for the pollination. Stigma is protected from the contamination. So how it is protected? By doing emasculation. Emasculation is done only in bisexual flowers. That is removal of anthers from the flower bud before the anther dehisces, break होने से पहले. Bagging. Flowers have to be covered with a bag which is made up of butter paper to prevent unwanted pollination या contamination. When the stigma of a bagged flower attains receptivity, the mature pollen grains are collected from the anther of the desired male parent and dusted on the stigma with the help of brush. And again, flowers are rebagged and the fruits are allowed to develop. So, ये artificially हम कर सकते हैं, but naturally that will happen. So, this is how pollination takes place. So, in next session, we will discuss a double fertilization and what are the results of double fertilization. So thank you for joining this session.